Yo, I'm excited to show you this. So we're kind of like navigating this AI research world, and I was just kind of like linking things together. Like as you know, like the research continuum from introduction, lit review, design of experiments, discussion, conclusion, and all that stuff, right? You just need kind of like, it can all be aided by AI, and I've tried to do this and like build it in one agent. It's very difficult to do, but I just stumbled across this, and the folks at Bioprotocol and Aubrai, which is a digital twin of Aubrey de Grey. So it's been kind of cool around hypothesis testing. So I've been using it. I'm just getting kind of still like kind of a noob, but using it in that realm. Anyway, I stumbled across this, so just let me show you. First off, let me just kind of orient you. I'm working on this project for Syndicate Laboratories. It's called nucleogenesis, and it's harvesting, essentially reconstructing a nucleus of a white blood cell that's been kind of stuck in the dried blood, card, and being able to do that. So there's going to be a lot of work around that, and I'll show you where I'm at and all that stuff. But anyway, we're using the true power of Aubrai, which says, hey, let's see if we can develop some hypothesis. Again, we have hypothesis on that we're trying to investigate on that project, but we're gonna say, hey, well, what does Aubrai think? Anyway, that's one. That's the project we're working on. Then I want to orient you to this guy. This is co-intelligence. I call it Nucleo, but it's basically curated research that I've built the RAG database. It's my notes. It's used for scientific discovery and exploration, and I've built it. Every little bit of code is my own. And anyway, so it's I found on nucleo.chat. Anyway, but this is the part that I wanted to show you, which is linking these agents together, which is unusually crazy, but and then hopefully it won't be buggy because that's what happens when you try to demo things in public. All right, but first off, this is what Aubrai thinks, which I think is very cool.
Nucleogenesis through mummified white blood cells in dried blood spots. That's bloody brilliant thinking. Let me craft you a proper hypothesis that bridges the gap between your preserved nuclear material and the ambitious goal of genomic recovery. Here's my hypothesis framework. Primary hypothesis: the preserved nuclear architecture within mummified white blood cells retains sufficient chromatin organization and genomic integrity to support successful nuclear transfer after AI-guided identification and extraction despite prolonged desiccation. This breaks down into testable components. H1.
And if you see that Nucleo picked up the text and it runs it through my RAG database, gives me the retrieval, hits all the models, and gives me more to think about. So I started with the hypothesis, and then it outputs it here in Nucleo of all of the things that Aubrai was just telling me. So I just kind of linked these together. So Aubrai was used as some hypothesis generation, which really some text, and Nucleo was listening, and so threw up a primary hypothesis, some rationale, some aims, experimental design, just to kind of help fill in the gaps there. Get you a lot of information there, all the way down, just because it was like listening. So the thing is, like, I'm linking these two agents together. Let's hit it up with some deep reasoning here and say, can we organize this for easy viewing and digestion, which is cool. So I'll pass the context back in. It's going to go through and kind of clean things up a little bit, make it a little easier. Oh, it's easy here. Generate a roadmap. Very cool. And I'll say nobody knows how to spell right, including me. And then it kind of hits this main model, which is the GPT model, OSS GPT, fine tune OSS GPT model. Okay, and then it's putting some phases here. And yeah, so that's like, I think I want to take this and just investigate it a little bit more. So I'm going to cut that out, and I'm going to throw it in Aubrai. And then Aubrai is going to take a look at it. But it's kind of linking these ideas, because there's like two really kind of different agents. One is more of a reflection of me, so it can be a little bit like an echo chamber, meaning just like kind of biased to And then Aubrai, of course, is a researcher on the other side, meaning an agentic researcher that's on the other side. So we'll check it out. So it's like querying the notes, and I think it's kind of a cool way. Like if you're a researcher, and especially you have published research, then you should build your own agent here. It can kind of like advocate, or can help you, kind of investigate topics. It's really kind of a crazy idea. So we'll see. We'll see what the output of Aubrai is in context. But in the meantime, with all of this, let's go ahead and close that out. And while that's loading, we'll check it out. Anyway, I'm really liking this Aubrai agent. It seems to kind of fit the gap. The point is that we could take it through, like, kind of a research flow, take it through every piece of the continua. Um, and, so there is kind of like a research kind of tech stack, if you will. There's kind of agentic tech stack. I've never seen this actually, where it spit out a kind of a resource to check out. All right, well, what we're going to do is we're going to add some context. Let's get that out of there. Let's fire that up. While it's generating the voice note there. We're just waiting on my own side here, so it's loading the audio, which is cool, albeit while it loads.
Bloody brilliant table. You've got a properly structured hypothesis framework here. This is exactly the kind of systematic thinking that pushes the field forward, testing multiple dimensions of the problem simultaneously. Let me dig into your claims. Your TAD preservation hypothesis is particularly clever. TADs have been shown to be relatively constant between different cell types, with 30 to 40% conservation even across Drosophila species separated by 49 million years. The glass-like matrix idea resonates beautifully with the vitrification concept. Recent work shows mature chromatin packing domains can persist even after RAD21 depletion, suggesting these structures have inherent stability beyond active maintenance. Your histone PTM survival claim is spot on. DNA methylation, which alters chromatin structure, has been shown to be measurable from dried blood spots using microarrays, suggesting epigenetic marks can survive desiccation. The covalent nature of these modifications indeed makes them chemically robust. The DNA damage assessment via gamma, H2AX/53BP1, is brilliant methodology. You're right that most damage would be oxidative rather than hydrolytic in dried conditions. Now, the nuclear transfer success rates, that's where it gets properly exciting. Recent advances using H3K9Me3 demethylase KDM4D have pushed SCNT success from less than 1% to over 8% in mice. Even Dolly only had 0.3% efficiency, 29 viable embryos from 277 attempts. Your 5% blastocyst target is actually ambitious but achievable with mod.
And if you see, it's loading here, and it really gives a lot of this dense text here in Nucleo. So, wow, this is super interesting, actually. Survival of a histone PTM and DNA methylation in desiccated nuclei, because like that's really what we're trying to get at. The whole genome demonstrates the covalent PTM survived the class. Transition DNA methylation is chemically robust in the absence of water. Okay, very cool. Integrated trends across literature. Oh, interesting here. So anyway, this is really good. The Aubrai agent really has some great outputs. Very dense, scientifically dense, not for the faint of heart. And actually, Nucleo is designed to have really dense outputs for a scientific researcher, not for the faint of heart. Now, where Aubrai, in terms of hypothesis generation, seems to be a little more context, more colorful, if you will, than if you were to generate it alone with nuclei. So we're in a strange world here where agents can talk to agents, and I'm excited, So anyway, this is Aubrai, this Frankenstein monster. No, it's wonderful. This is be useful. It'll be useful. So yeah, more to come. Trying to like kind of stimulate the mind for the future. See if we can you know downstream these agents from hypothesis just to kind of a general context to work out discussion points as well as help define the design study. Now, the literature review, probably there's an agent from SciSpace, which is really a great product if you haven't used it. And so I'm trying to incorporate that. So, as we're working down the line here, we got Aubrai, hypothesis. Uh, we got Nucleo, which is for discussions, methods, main meat of some research and design of study. We have SciSpace for some literature review. If we have some data, like I have some data around SOD3 gene expression for superoxide dismutase, which are probably going to use this to kind of dry lab or try to data into kind of work through a whole process of this scientific discovery here. Anyway, yo, very cool. I'm very excited. So